This summer, my partner Renee Sarpong and myself, Jody Mizador, worked alongside our advisor, Dr. Kristen Bliss, on an analysis of independent and combined effects of elevated carbon dioxide and temperature on major agricultural crops, Solanum tuberosum and Solanum lycopersicum.
As we study trends, it is important to note that atmospheric carbon dioxide concentration has risen by more than 45% since pre-industrial times. From equilibrium levels of about 280 parts per million in 1880 to the currently observed levels of 407 parts per million. This dramatic increase is a direct result of human activities, primarily things such as fossil fuel burning, cement production, and modified land use patterns. If present emission patterns continue into the future, atmospheric carbon dioxide will be double by the end of the 21st century. It is suggested that the projected increase in carbon dioxide will modify the global climate by causing widespread rising of surface temperatures as well as air temperatures. This is done by the alteration of precipitation patterns and global hydrologic cycle, which increases frequency of severe events such as droughts and flooding. Agricultural crop production is one of the key human factors that may be significantly affected by this, with consequences to global food supply. Ultimately, the net effect of increased carbon dioxide and temperature may be detrimental to agricultural crops. Tomatoes and potatoes were selected because they are included among main horticultural crops worldwide. Due to their broad availability and nutritional characteristics, they are considered to be two of the most important components of the human diet. Nevertheless, potatoes and tomatoes contain a naturally occurring toxin called solanine, which comprises a great amount of the total glycoalkaloids present in the plant. Solanine is a physiologically active glycoalkaloid of these two plants, along with other species of this genus, such as eggplants, tobacco, peppers, and a lot more. This alkaloid is bitter tasting and has poisonous properties that have caused concerns in certain geographical areas. Symptoms of glycoalkaloid poisoning include cold pain in the abdomen and stomach, vomiting, fever, rapid pulse, low blood pressure, and neurological disorders. In this experiment, we increased the level of carbon dioxide and temperature in potatoes and tomatoes to determine what, if any, detrimental effects occurred. We expected to see an increase in biomass due to increased carbon dioxide, but we also expected to see that increased temperature might decrease biomass. We speculated that increased carbon dioxide and temperature in combination might increase toxic solanine levels in both potatoes and tomatoes. By analyzing the biomass, leaf angle, number of leaves, height, and solanine levels, we will be able to see if the factors set by the experiment, which mimics factors of climate change, have an effect on these agricultural plants.
We started this experiment by choosing agricultural crops, tomato and potato. We then found the environmental conditions to replicate, being elevated CO2 and temperature. We chose between 280 and 407 for ambient CO2 and roughly 1,000 parts per million for elevated CO2. We chose between 80 degrees plus or minus 5 degrees for ambient temperature and 90 degrees plus or minus 5 for elevated temperature. We then set up environmental chambers that would mimic the atmospheric conditions in each of our locations. We chose tubes to use for administrating CO2, heating pads which will simulate increased temperatures, and CO2 sensors to detect the CO2 levels in the chamber. Each chamber would mimic an atmospheric condition. One chamber would have ambient CO2 and elevated temperature, elevated CO2 and ambient temperature, ambient CO2 and ambient temperature, and elevated CO2 and elevated temperatures. We monitored the plants for 3 weeks, attending to the plants when needed. At the end of each week for 4 weeks, we collected measurements of the length, width, amount of leaf, and other morphological characteristics. At the end of the third week, we harvested above ground samples for biomass and obtained final measurements and recorded leaf angles.
To begin, we used a t-test, which is a statistical analysis used to determine if there's a significant difference between the mean of each treatment group compared with the control. We started with the measurement of biomass, which is considered a good measurement of plant health because it reflects the amount of sunlight, water, carbon dioxide, and minerals a plant is able to capture and turn into plant mass. The t-test described the trends as not being significantly different. The trends were similar in tomatoes and potatoes, with control plants having the lowest biomass. Elevated carbon dioxide alone, elevated temperature alone, and elevated carbon dioxide and temperature combined all had higher biomass than control plants. The difference were not significant due to the large differences in plant across all three replicates. We also measured leaf angle, which was shown to be significantly larger in plants with elevated carbon dioxide, even regardless of temperature. Although these morphological characteristics showed trends other than leaf angle, they were not statistically significant, and the main goal of our study is to analyze the chemical components of this plant to determine if the toxin solanine would show any significant trends, specifically in the plants with elevated carbon dioxide and temperature. Due to the restrictions set by the current global pandemic, we were unable to conduct the portion since it requires access to a laboratory. We had planned to do this bulk of the experiment when we were allowed back and able to access the resources needed for this.
In this project, we examined the combined and independent effect of enhanced levels of carbon dioxide and temperature on plant growth. Increases in carbon dioxide and temperature had a profound effect on the two major crops that we studied. In tomatoes, all treatment groups had higher biodiversity than controlled plants. Therefore, in tomato plants, our data showed a trend towards increased biomass of increased carbon dioxide in spite of extremely warmer temperatures. Perhaps the plants benefited due to the increase in leaf size. An increase in leaf size could provide more leaf surface area, which would promote photosynthesis, thus increasing biomass. We need to interpret our results with caution, though, because the trend that we observed were not statistically significant except for leaf angle. Overall, our ultimate goal of the study was to assess and determine the impact of these components on tomatoes and potatoes. Some of the restrictions that we faced during this experiment was due to the COVID-19 pandemic that restricted us from many of the resources that we would have originally obtained by the Randolph campus, such as a well-ventilated and operating greenhouse, as well as the faculty and the proper equipment. We also had some financial restrictions as well. There are many factors that determine the response of crops to changes in climate conditions and to elevated CO2 concentrations. Therefore, in the future, we would like to standardize replicates and increase replication, quantify the chemical properties of toxins in the plants, identify health implications, monitor and observe nutritional properties within each treatment group, and assess climate change impacts on agricultural industries. This brings us to the end of our presentation. My partner, Renee Sarpong, and myself, Jody Mizador, would like to acknowledge the following people for their assistance and support throughout our research. Thank you.